DNA is a nucleic acid made of nucleotides joined into long strands or chains by covalent bonds. The monomer of nucleic acids is a nucleotide. They are long chains that are somewhat acidic. Nucleotides include three basic components: a 5-carbon sugar molecule, a phosphate group, and a nitrogen base. Nucleotides join together to form strands of DNA. The nucleotides that make up DNA have four types of nitrogenous bases: adenine, guanine, cytosine, and thymine. Each of these is often referred to by its first initial, A, G, C, or T. Covalent bonds connect the sugar of one nucleotide with the phosphate group of another nucleotide, and these can join in any sequence. DNA is a nucleic acid made of nucleotides joined into long strands or chains by covalent bonds. Biochemist Erwin Chargaff discovered similarities in the percentages of bases in DNA. He identified that each sample of DNA included an equal percentage of adenine and thymine, as well as an equal percentage of guanine and cytosine. This realization created what is known as Chargaff's rule: A equals T and G equals C. Scientist Rosalind Franklin used X-ray diffractions to study the structure of DNA molecules. After stretching the DNA fibers to make the strands as parallel as possible, she X-rayed the samples and recorded the patterns they created. Although she was not able to fully determine the structure of the molecule, her work provided insight into the helix shape of DNA strands. James Watson and Francis Crick were studying the structure of DNA at the same time as Franklin. Although they were able to build three-dimensional models of DNA, they still could not explain its properties. After seeing Franklin's X-ray of DNA, they determined that its structure was that of a double helix. The double helix model explains Chargaff's rule of base pairing and how two strands of DNA are held together. The two strands of DNA's double helix run anti-parallel, or in opposite directions. The structure connects the nitrogen bases on each strand and allows DNA to carry nucleotides in a specific sequence. DNA strands are held together by hydrogen bonds formed between nucleotides. Nitrogenous bases bond with certain other bases in a process called base pairing. For DNA, adenine bonds with thymine, and guanine bonds with cytosine. Base pair clarified how Chargaff's rule applied to DNA and why A equals T and G equals C. This led to a Nobel Prize for Watson, Crick, and Franklin. Although base pairing explained DNA's structure and sequences, it did not explain how DNA carried or used the genetic information.